a great mentor to me, and I know a lot of others through all these years. And I just feel really honored to be able to introduce you. As you know, we go through and we talk about people's awards and honors, and I think most of us will say 2020 was not necessarily a year that we look back to with a lot of good feelings. But looking at your list of awards, I noticed you had quite a year in 2020.
Yes.
This invitation is based on Veronica being the 2020 recipient of the ACS, American Chemical Society's Irving Langmuir Award. But also in 2020, Veronica received the American Institute of Chemists Pioneer Award. She became a member of the National Academy of Sciences and received the Wilbur Lucius Cross Medal from the Yale Graduate School Alumni Association. So somehow you managed to get all the good things happening in 2020, which is really quite amazing. There's a number of other awards, but I think that gives a pretty good sampling of the various types of awards that you've won. In terms of science, what I just absolutely adore about what Veronica does is that she uses spectroscopy, various types of spectroscopy, but usually vibrational spectroscopy, to ask and answer really big questions, primarily in atmospheric and environmental chemistry, but also in terms of questions about the origin of life and the generation of peptide bonds that are just absolutely critical to go and start making amino acids and other biomolecules. So it's a great pleasure to introduce Veronica. This is part of, I think, your official Irving Langmuir Award lecture, since that never really happened in 2020, entitled Water-Air Interface, Neutral Reaction Environments. And before you get started, and as you're pulling up your presentation, I want to remind everyone that the way we'll do this is we will collect questions through chat, and then at the end of the presentation, we will curate the questions and ask them on people's behalf. After the question and answer session, we will ask students and postdocs to stay on if they like to interact a little bit longer with Veronica. So I'm looking forward to your talk, and it's all yours.
Thank you. Thank you, Anne, for a very gracious introduction. With that, let me see if I can manage to share the screen. Okay, so indeed, 2020 was a big year in a way, but of course, all the celebrations that went with all these awards got to be remote or canceled. So it was an interesting year, as you all know. It is my pleasure and honor to give this talk. And before I start, I wanted to acknowledge as well as dedicate this talk to Irving Langmuir and to Kathleen Blodgett, the two of whom worked together at GE since 1917. My work, the work I'll talk about today, which involves chemistry at aqueous surfaces and interfaces, is enabled by their work. As Anne said, this talk is prompted by the ACS Irving Langmuir Award in Chemical Physics, which is to recognize and encourage outstanding interdisciplinary research in chemistry and physics in the spirit of Irving Langmuir. Furthermore, I wanted to mention that Langmuir won the 1932 Nobel Prize in Chemistry for his discoveries and investigations in surface and surface chemistry. And that's a work that I will build upon. I was the recipient of the 2020 Irving Langmuir Award, and that was for contributions revealing fundamental organic photoreactions at water interfaces in planetary systems, including the contemporary and ancient Earths. And so now getting to my own work, I wanted to start by mentioning what the inspiration was for my group to retool about 20 years ago, to retool and start looking at aqueous surfaces, interfaces, and organic chemistry at such interfaces. The inspiration came through measurements of a single aerosol particle composition, which showed an organic content that persisted even at high altitude. And at the time, the expectation was that since these organics mostly are insoluble, they would not be found on aerosol particles. And so in response to that, or thinking about this, with my friends and colleagues, Barney Allison and Adrian Tuck, we developed a model that looked at these aerosol particles as saline solutions encapsulated by an organic film. And to that, another friend, Chris Dobson, contributed to adding another dimension, which was to think of these aerosol surfaces as important chemical environments. So at this point, it was obvious that what we really need to know, and not enough of the chemistry at such surfaces was known, was what happens in these organic films at water-air interfaces. And so as a result, I developed some laboratory capabilities that instead of using molecular beams and jets as we have done, and now use Langmuir and Langmuir Blodgett troughs, as I will show you in a second. So why would this matter? Well, in the consequences in the contemporary atmosphere are the fact that aerosols influence climate through absorption and scattering of incoming radiation. But scattering of solar radiation depends on the size of these aerosol particles, on the hydrophilicity and ability to nucleate clouds. And so all these properties and changing of the aerosol surface itself are relevant in this context. And on ancient Earth, and that's certainly would be true on other planets, because aerosols would form on any rotating planet with a liquid ocean. They have a very large surface area and could have provided prebiotic reactor for biopolymer and protocell formation. And so what I mean by this is that in the absence of biology, the question, which is sort of the same question we ask for the contemporary atmosphere in a way, is are there processes that can happen at the surface in the absence of biology, not requiring enzyme catalysts that lead to biopolymer formation? So what I will do is illustrate some of the work we are doing with chemistry and photochemistry at the water surface, and choosing problems that would be relevant to the natural environment. So we look at the analogs with water interfaces, ocean lakes, atmospheric aerosols, and fog and cloud droplets. So I'll give us 3 examples. One is the amide bond formation, and that would be in the dark. It's a reaction at the surface of water, but not, we haven't studied the photochemical version of this. Then the second example does use light and forms oligomers, building complex supramolecular structures and biopolymers. And just briefly, I'll mention some work we did with vesicles and cell membranes that are sort of following up on complex structures that we can make. So we use in our work both flat surfaces, and that's where the Langmuir blodgett troughs are the tool of choice. We use surface sensitive spectroscopy as well as analytical methods like mass spec, NMR, and infrared spectroscopy to identify products, as well as follow the development of the chemistry in time when we can. We also would like to look at aerosol surfaces. And so in a collaboration in Paris with Jean-François Dussin, we use atmospheric simulation chamber where we generate nebulized drops and look at the chemistry that happens on and in these drops. So while our work is fundamental in nature, and any of the meaning that we would extract would not be possible without connecting these lab findings with results of field measurements. So I do keep an eye on, and I'm quite interested in developments in that field. So let me tell you a little bit of how we do this. So there is a Langmuir trough. What you see in the picture on the left-hand side is the trough itself. We prepare a solution, an aqueous solution of whatever composition we want. And then either place an organic surfactant at the surface of it or have an organic dissolved in the solution, partitioning to some extent to the surface. The two barriers can move in, and the Wilhelmy balance measures the surface pressure, which then can give us information, thermodynamic information about the surface tension, and so on. It also can be used, the trough can be used to control the samples so that when we go on and look at chemistry or follow a reaction, we can control the state, the proximity of these molecules at the surface. We've recently added the blodgett tool to the Langmuir trough, which allows us to dip a window in and out of the solution and allows us to collect samples from the surface of water as we collect at the same time sample from the bulk. And then we analyze the two separately to look for differences and similarities. So in a way, our surface information has an internal standard. We've also extended our setup, or this Langmuir blodgett trough, with a bank of LEDs that sit above the trough that match the profile of the trough. And so we can do photochemistry as well. The other tool I mentioned that has been a lot of fun to work with is called Sésame, and it's an atmospheric simulation chamber at University of Paris Est at Créteil. This was designed and perfected by Jean-François Dussin, who is the guy in the middle of the picture in the black shirt. The chamber itself is in the background. It's a 4,000 liter stainless steel chamber. It has many ports, and you don't see because there are these Kevlar skirts at the top, which contains 3 gigantic solar simulators. And so it's a very, very nice photochemical chamber, and it allows us to look at gas phase, or we nebulize drops. So these are now experiments where we can look at drops of a certain size and use FTIR as well as PPRMS, as well as measuring particles to obtain information about the progress of the chemistry or photochemistry. So in this picture, the woman in the front is Alison Reed Harris, who was a graduate student in my group, now has finished and gone on to better things. And the rest of the people in the picture are part of the French team that helped us do these experiments. So I wanted to mention that we will use spectroscopy, and there is a lot of very beautiful work, both theoretical and experimental, using nonlinear techniques. And so vibrational spectroscopy has been done elegantly with such tools. And I'll mention some of that. Also wanted to mention vibrational spectroscopy of solvated ions. But what we are doing is sort of exploratory. I think it goes to look at chemistry, and we don't always know what we are expecting. So it's very hard to obtain and analyze these using nonlinear techniques. So we are going to use surface reflection, and primarily I'll talk about infrared reflection from the surface of water, even though we now have the capability of looking at UV surface reflection as well. So this has a technique that is easier to use in some ways. It doesn't have the sensitivity of a nonlinear techniques, but it is, when possible, it is very easy to interpret, and it lends itself to finding new features that perhaps we didn't suspect. And I want to thank Heather Allen for suggesting doing this. So what are we doing? We're looking at primarily at IR reflection or iris, and you can see actually both a diagram and an actual picture of our setup, which is external to the FTIR spectrometer. So the radiation from the FTIR spectrometer, the infrared radiation, is brought into the chamber. You see a little trough in the chamber and mirrors that both steer the beam to the surface and then collect the reflected light. And sort of that's the experiment. We then get an experiment from the surface. I wanted to mention that we're still learning a lot of things about this technique and making changes as we speak. But I wanted to mention the differences between this reflection absorption spectroscopy versus ATR. We use both. We take ATR spectra, which give us a good idea of what the infrared spectrum may be of our compound, even in aqueous environments. But it's not a surface sensitive method, as opposed to the reflection absorption spectroscopy, which comes from less dense medium, air, and reflects off the monolayer, and the reflected beam then gives us information about the surface. And at any rate, a lot of credit goes for the figure as well as for developing some of these techniques to Benjamin Frenzen, who's a postdoc in my group. I wanted to show you some new results that have not yet been published. They are part of Lexi Deal's PhD work. And she looked at lactic acid and hydroxy acids, which was different chain length. In this version that Benjamin Frenzen had implemented, we actually use polarized light. The S-polarized light interacts only with the in-water plane, and the P-polarized light interacts with all the components. And so the figure furthest to the left, you can see these are all S-polarized spectra. You can see the one of lactic acid and hydroxyoctanoic acid, and not much from this is the C double bond O stretch, not much that we can see in the hydroxysteric acid, which is a monolayer. And then the CH region, CH stretch region, that one you see very clearly in hydroxysteric acid, which has such a large number of CH2 groups. And on the furthest image, you see the P and S polarized spectra, which the P polarized spectra are positive, the S polarized features are negative, but they'll give us information on how these molecules sit with respect to the surface. And so this is the setup is quite new, and we are only beginning to explore it. So I'll turn to the first example, chemical example I wanted to talk to you about. And these is chemistry at the water surface. And what I mean by that, it's chemistry that actually does not occur in the bulk. It's chemistry that's quite important in biology. And of course, enzymes do it these days. Part of the interest is in forming oligomers, but it's going to be also important in prebiotic chemistry. The peptide bond falls in this category, the glycosidic bond and phosphodiester bond formation. All these involve the condensation of precursors of the monomers and the elimination of water. So these reactions are not favorable in water. The equilibrium constants are pitifully small. And what's favored is the hydrolyzed products. And so I'm going to show you our attempts at forming the peptide bond with the assumption that the surface would be a much more auspicious environment to do this chemistry. And so, so again, it's a condensation reaction. We are going to take, we're trying to make the peptide bond, and are based on the assumption that I think is fairly reasonable, that there is an asymmetric solvation at the interface, that while the molecule has access to all the monomers that live in the aqueous solution, it's also can eliminate water because of the solvation is only partial. And we're going to use a metal salt for reasons that have to do with literature suggestions. I wanted to mention there was some work done by John Oliver, who was then at Brown, that used amino acids, synthetic amino acids that was very low on carbon chains that were absolutely not living in the water, and could perform this chemistry, and used a copper salt. So we are using now soluble amino acids, and furthermore, we're going to use the amino acid ester. So the picture on the left shows you the iris spectrum, and if you see the negative going peaks that are about 1700 wave numbers, that corresponds to the C double bond O. And at around 1,000 wave numbers, that feature corresponds to the amine twists. when we add the copper salt, the copper does bind to the amine part of the molecule, and we think it has performs the role of bringing more of the molecules to the surface. So after a few hours of doing absolutely nothing, this is an entirely in situ experiment. We just keep taking iris spectra. You see a feature that grew in at a little over 1600 wave numbers. That's in the red block. That happens to be the amide 1 band of a peptide. And we don't know from our experiment at this stage how big these complexes are. We know the bonds are there, but we don't know much about their structure. However, this chemistry would not happen in aqueous bulk, so we were very pleased to see it happen at the surface. Now, peptide bond formation is complicated by another factor, and that is the fact that amino acids are actually zwitterions. And they're zwitterions, well, this to form the peptide bond, you're working with the unionized species. So I won't have time to tell you about this, but we did look at a range of pHs at phenylalanine, another amino acid that has some propensity for being at the surface. And we looked at the state of ionization at the surface by measuring the vibrational spectra through this technique. And in parallel, we extracted from the bulk at the same pH a sample, and we measured the pH there. And we found that at high and low pH, there was more propensity to have the unionized species. And so that would favor the chemistry as well. So in cartoon form, I wanted to mention that such chemistry, and again, it's not only peptide bonds, there are lots of esterification reactions, amide bond formation, many others that require condensation and elimination of water. Such reactions would occur. They would also occur at the sea surface microlayer, but I want to point out that the surface area, let's say on Earth, of all the aerosols is far greater by orders of magnitude than all these flat surfaces. So probably a lot of the chemistry would occur on aerosols. In addition, the aerosols, the pH, the salinity, many other characteristics are changing, and so provides a range of chemical environments, always with the underlying property that the reagents are concentrated and water can be eliminated. I'd like to now change to my second example, and that has to do with now light, and specifically we're going to consider sunlight, but actually starlight if you happen to be on a different planet. And the question is, can you get, or how can you get energy transduction from solar radiation and put that energy into high energy chemical bonds? And so this chemistry, we hope, will then lead to building chemical complexity to generate the biopolymers required for life. So in this study, sunlight is the energy source, but water, specifically the water-air interface, is the preferred reaction environment. So just a couple words. We are quite aware that solar radiation is not was a bit different in prebiotic times. I like the work of Ranjan and Saselov in that respect, where they give some modeling studies that tell us more about what radiation was available. In the top panel in red is a modern solar spectrum outside the Earth's atmosphere, and in blue is the solar radiation that actually reaches the Earth. So you see it's missing some big bites that are due to absorbers such as water in the infrared, oxygen, and then ozone in the UV, into the high energy end. And while it's not entirely known what possible shields there would have been, there was no oxygen in the prebiotic Earth, and so no ozone. So the radiation would have, more infrared radiation would have been available. All this to say that we are perfectly able to do this chemistry by paying attention to the excitation that we provide. And so to illustrate this, I will turn to oxo acids to start with pyruvic acid. And I'll tell you that it's a terribly interesting molecule. It can be synthesized and has been synthesized abiotically. And so it would have been in prebiotic Earth. It's also found in meteorites. So we know it's being synthesized somewhere, likely abiotically. It's an old currency for metabolism, serving as an intermediate in both anaerobic and aerobic metabolic pathways. And so we looked at its chemistry and photochemistry in gas phase, in the ground electronic state, where we drove this chemistry by OH vibrational overtone excitation, and also in the excited electronic state by exciting the UV, which is between 300 and 400 nanometers. So we were able to look in at the multi-phase reactivity of pyruvic acid, namely gas phase, aqueous phase, and discover the surfaces and the surfaces of aerosols or aqueous solutions and interfaces generate different sets of products. We are specifically interested in oligomer and polymer formation, and found that that these polymers and oligomers are found then at the surface, and therefore they would lead to basically modifying the aerosol surface as a result of this photochemistry. Very briefly, in gas phase, photochemically, there was some quite a bit of work. As I said, we also did the ground state and found that the product is metal hydroxycarbene, which is formed through a hydrogen atom chattering mechanism, thanks to Rex Koji for thinking about this. And so the main products end up being acetaldehyde, but also under atmospheric conditions, we see acidic acids that proceeds through a different mechanism. The situation when you have an aqueous solution is very different. And that has many reasons, but one of them is that even one water molecule can catalyze reactions, and specifically aldehydes and ketones do hydrate to form diols. And so then we did look at some of this chemistry with clusters and very small number of water molecules. and as well as in aqueous solution. So specifically, pyruvic acid can form diol, and so we became aware of the fact that when you look at the solution of pyruvic acid, you have always 4 species. Pyruvic acid in its keto form, as well as the diol form. It's depending on pH and such, it's about 60-40% favoring the diol. And then in each case, you can get the anion. And so really, under these different conditions, you have a different mix. You don't have one reagent, you have 4 species that you have to keep looking at. And so here I need to give thanks to Barry Carpenter, without whom we wouldn't have been able to write all these beautiful mechanisms that take pyruvic acid and in aqueous solution, they form the triplet state, which then hydrogen abstracts, and you end up with radicals and the follow-up radical reactions. The main product I want to point out is dimethyl tartaric acid. And that's going to be true of pyruvic acid as well as the oxoacids I'll mention in a second. And furthermore, my group was the first to actually look at high pH. And interestingly, then at high pH, because you have the deprotonated from the anion, you abstract hydrogen from the CH2 group. And we can identify several of the products which can be different. So at high pH, you get DMOHA in a larger quantity. So the ratio of dimethyl tartaric acid and DMTA and DMOHA helped us sort out the chemistry at low and high pH. This is just a feature spectra that we obtained in the simulations chamber sesame through mass spec, PTRMS. And I wanted to show you that that's actually how we found that the green trace, which took a while and definitely help from Barry Carpenter to identify, zymonic acid is something that we never saw before in aqueous solution, but we did see it in sesame, where we had lots of surface area of aqueous drops. So acetic acid is the orange trace, and the blue trace is acetaldehyde. So the emphasis, as I mentioned, is on these oligomers that form preferentially at the surface. This is a very recent study, Keaton and Lexi and several helpful collaborators. And I just wanted to show you the negative mode ESI mass spec. The bulk is shown in red, and the black is what we collected at the surface. So these are experiments where the bulk and surface samples are collected simultaneously. And the interesting thing is that some photolysis products, like this DMOHA and the DMTA, dimethyl tartaric acid, are found primarily in the bulk. However, zymonic acid, and we see lactic acid, and oligomers of pyruvic and lactic acid are found primarily at the surface. So there is a very different preference for surface partitioning of these. And so the dark chemistry, we proposed mechanisms, and want to again thank Lexi and Keaton for this, that we can have mechanisms for how we might form in the dark zymonic acid, as well as this pyruvic and lactic oligomers, as well as forming the lactic acid photochemically in solution from pyruvic acid. I would like to turn to using the exact same chemistry I just told you about, but this time using a close relative of pyruvic acid, 2-oxooctanoic acid, which has a 6-carbon tail. So it has more of a preference to be at the surface. And the chemistry in a nutshell requires sunlight, requires water, and this oxo acid. And the result is the major product of this chemistry is forming a carbon-carbon bond and double-tailed surfactant. Not only that, but we've shown that if you have a mixture of oxo acids, you also get the mixed products. And perhaps the most interesting scenario was that when an oxo acid can activate a fatty alcohol or a fatty acid that themselves do not undergo photochemistry in the solar spectrum, but activated by the oxo acids, they do form these multi-tailed surfactants. And by the way, these were interesting for origin of life scenarios because they're sort of a new class of lipids. So Becky Reff drew a picture that shows just some of the multi-tailed surfactants that came out of her photolysis of 2-oxooctanoic acid. And there are a lot of these that then we could show using the Langmuir Prof have a lot higher propensity to be at the surface. So the image shows in red the 2-OA solution before photolysis. It does have some surface partitioning, but it's increased immensely as a result of this chemistry when we have all these lipids basically at the surface. So then it turned out this was a serendipitous event where Elizabeth Griffith, who was doing these experiments, wanting to show that sunlight can be an agent for building complexity, partly as a response to the origin of life community that thought of sunlight and UV photochemistry as being destructive, but hadn't actually contemplated the fact that the radicals generated photochemically can lead to polymer and oligomer formation. At any rate, Elizabeth was repeating these experiments, she saw after a few hours of photolysis that the sample became cloudy. So something was happening spontaneously to our sample. We looked at the microscope image and found spherical objects, also dynamic light scattering, then were able to tell us that they are relatively similar in size. There is follow-up work that's very nice work with time result, dynamic light scattering and imaging that's done by Kevin Wilson at LBL. Anyway, these, we still don't know what these objects are. They look, but we know that they're spherical and that they are they are of similar size. So we've just generated not only a more complex molecule, but a spontaneous supramolecular um, um, object. So I'm just showing some of the possibilities, and we pondered, what these objects are that we made. 200 nanometer diameter, they are too big to be micelles. We thought they would be vesicles, but they likely are something different altogether. However, the molecules themselves are a new set of completely prebiotically acceptable, relevant lipids that may have existed before, only requiring sunlight and available precursors could have existed prebiotically. With that, now that I've reached the point of, okay, can we have enclosures and cells and so forth, I just wanted to briefly mention some work that we've done with vesicles and membranes. I want to thank Sharif Mansi for helping us, teaching us how to make vesicles and how to study them. The work I'll be showing, it was done by Russell Perkins when he was a graduate student, and basically is looking at a problem that's interesting for medical reasons, and that's phenylalanine. Phenylalanine can form phenylketonuria, which is a pretty awful disease. All infants are tested to their ability to metabolize phenylalanine, because if they can't, it's a bit of a problem. It looks like phenylalanine destroys the membrane of cells and causes all sorts of side effects, including brain damage. And so Russell did make models for cell membrane using DPCC, or 1,2-dipalmithyl glycerol 3-phosphatidylcholine. And these are some of the images that he got of these vesicles that had or didn't have phenylalanine. So the experiment showed that he did that phenylalanine has the ability to alter the permeability of bilayer vesicles, which we used as model membranes. And it appears that the ability to do that has to do with phenylalanine aggregating at the interface and forming basically channels in this membrane. And so using the dyes and fluorimetry, Russell was able to look at, as you see in the picture on the bottom right-hand corner, to look at what happens, how permeable is this model membrane to phenylalanine. And indeed, the membrane became much leakier as the concentration of phenylalanine increased. And here I owe that to Annette Erbsey, who taught and worked with Russell on those fluorimetry experiments. So I'm coming to conclusions, and wanted to just step back from what I've just said to show you that these are examples that I hope show that sunlight and water are agents of chemical complexity. Water surfaces, as in sea surface or lakes or aerosols or cloud droplets, can sustain and promote organic reactions that are not allowed in bulk. Peptide bond formation, for example. The Zaire group has seen a phosphorylation of sugars on a microdrop and so on. We've also looked at sunlight and photochemistry, solar photochemistry of oxoacids. And we've seen that you can generate polymers which live or partition to the water-air interface. And that this chemistry does provide a non-biological source of chiral molecules as well as aggregates at the sea surface and in sea sprays. So the big main conclusion is that we can build chemical complexity in the natural environment at the surface of water. And we've used that both to explain what might happen in today's atmosphere as well as in prebiotic scenarios. So that's a cartoon version of where such interfaces are in the natural environment. We've looked at flat surfaces, and we can play around with the salinity and pH and so forth. pH of the ocean is about 8. Then atmospheric aerosols become acidified, and so they have a changing range of pH. They also they become more concentrated in salt, they coagulate, they divide. There are a lot of interesting additional processes. Eventually, some of these return to the ocean, we've also looked at the properties of vesicles that may actually live in aqueous solutions. So with that, I'd like to mention that I owe a great deal to a large range of collaborators from many different areas. This work is quite interdisciplinary. We're learning all the time about planetary and Earth atmospheres and chemistry and various techniques. But Jamie Donaldson, Barney Allison, Adrian Tuck, Chris Dobson, Heikki Tervahatu, all contributed to conceptual developments of this aerosol encapsulated in an organic film idea. Sharif Mensi, who taught us about vesicles, Jean-François Dussin, and Jeanne Monod, with whom we worked in France on simulation chamber studies. Barry Carpenter was the key to us being able to write organic reaction mechanisms. Rex Koji, Henry Kiergaard helped with different aspects of the theory. Vicky Gracian and Heather Allen helped us do some of the chemistry at interfaces, including geophysical interfaces. The following people are in red are former group members. These are all people who have gotten their PhDs and have gone on. Nabila Rontu, Jessica Gilman, Elizabeth Griffiths, Alison Reed Harris, Becky Reff, Russell Perkins, Jay Kroll, and Dr. Sandy Bayer, who was a postdoc. They are no longer in my group, but I did talk about their contributions. Currently, the work is being done at a whole other level by Keaton Kappas, Lexi Beal, Benjamin Frenzen, and an undergraduate who now left for graduate school, Bree Hopper. And the research support came from NSF, from NASA Habitable Planets, and USARO. And with that, I'd like to thank you for your attention and happy to answer questions.
Thank you, Veronica. I guess we're going to start with the questions. Jahan Daulati has a two-part question. Is the role of the surface mostly in bringing the relevant reactants into proximity of each other and perhaps orient them? First part. Second part is, are other effects such as oriented electric fields arising from surface ion distribution can affect reactions akin to an electrochemical surface?
Yes, the answer is yes, yes, and yes. So indeed, at the surface, even soluble surfactants, and we worked with many of those, are preferentially come to the surface, maybe only 5%, as would be the case of pyruvic acid. But yes, you have a higher concentration. And there are orientational effects. So some of the spectroscopy we do inform us about those things. But then there are chemical effects, and that does include, there's some very interesting papers I didn't have a chance to talk about that are recently done in microdrops. So the Zaire group, Kevin Wilson's group, the Cook's group, that have shown that in microdrops, which are sort of just so happens the same size as aerosols and the same size as cells, that this chemistry happens at the surface and it's not thermodynamically allowed in the bulk. There is also, and that's true of condensation reactions, there is a thermodynamic issue. Even more intriguing, and we see some of these products at the water surface as well, but is the spontaneous redox reactions, where at the surface, presumably the electric fields involved help to overcome the redox potential. So you can do it without catalysts and without anything at the surface. So very interesting results that not just from our group, but especially I would like to call out this microdrop community that's been very active in the last 3 to 5 years to point to a number of different effects. Folding is also a different thing that can happen at the interface. So we are still learning a lot about what else.
Okay, thank you, Veronica, for the wonderful talk. This is Carlos Baez, UT Austin. I logged in a few minutes late, but so Marco and I will be alternating the questions. There are quite a few questions from the audience. So the next question is from Gernot Friedrichs, who asked, you mentioned that copper catalyzed amide bond formation does only take place at the interface, but not in the bulk. The question is, did I get this right? And if so, what is the mechanistic reason for this? And has it been verified by theoretical models?
The answer, I'll start backwards a bit. I think there is some theory being done now. Marie-Pierre Gaillot, for example, in France. But no, at the moment, I'm not aware of any theoretical models. It would be very interesting to see those. My understanding, and that is consistent with what we see spectroscopically, the copper really binds to these, in our case, amino acids. In the case of John Oliver, in our case, it's an ester, and brings them to the surface. But this chemistry does not occur in the bulk. The equilibrium constant is negligible. In bulk water, these guys like to be in monomeric form.
Thank you.
Yeah.
Okay, next question is from Sarah Keller. She says, you noted that you don't know what the structures are for your 200 nanometer features in the oxoacid mixtures exposed to light. And then the next slide of your talk, you show some cryo-EM images of vesicles. I know from experience that cryo-EM can be very challenging and expensive. But have you attempted to use cryo-EM to characterize those 200 nanometer features? And if so, what did you find?
Yes, thank you, Sarah. So we tried and failed. The images I showed were taken in Colorado in the cryo-EM, but those were vesicles we generated. They were fairly well-behaved, largish vesicles. But of these objects that self-assemble and so on, we failed to do it here. However, I've recently seen some very nice cryo-EM that Becky Reff, Kevin Wilson collaborated with Friedman, I think. And they appear to be phase separated. So they are spherical objects separated by phase, but this work is not published. Those images I've seen, but are not available. So there is work being done, and you're absolutely right. In principle, challenging, but possible. I think cryo-EM will shed light on what these objects are.
Thank you, Veronica, for the answer. The next question is from Stephen Ferber, who asked, related to your work on chiral molecules, actually. So the question is, are these chiral molecules possible sources for the overall chiral nature of biochemistry?
So we believe we have some ideas. We haven't done very much with the chirality. I think, I didn't show this, but I think what we are getting from an achiral precursor like 2OA is a racemic mixture. However, at the surface, you immediately have a preference for one, in this case would be the meso conformer. But what we would like to do, we haven't so far even started to do it, is to look at what happens on self-assembly, whether you could end up with preferentially chiral structures. But there are atmospheric measurements, and Franz Geiger has published some intriguing papers showing chiral molecules in these aerosols. While they could be of biological origin, we'd like to suggest that there are also abiotic ways in the atmosphere that produce chiral molecules. So we just haven't done enough work along those lines, but I think there are some interesting possibilities.
Very interesting. Thank you.
The next question is from Rudy Marcus. It says, Betty Sharpless has obtained evidence that the dangling OH bonds at the water interface can catalyze certain organic reactions. We believe those are the reactions where the transition state is more H-bonded than the reactants. Have you seen such behavior in any of the reactions you have studied?
Yeah, that's very interesting body of work, and I'm aware of it. We haven't been able to identify the transition state in that way, but I think it's quite possible that these dangling OH bonds would play a role in some of this chemistry. I think the examples we had appear to be primarily like the condensation reactions, sort of driven by the thermodynamic, a different thermodynamic system at the surface. But it's a great suggestion. I should look at our chemistry to see how the hydrogen bonding, which is very important. For example, all these oxoacids have an intramolecular hydrogen bond, which gets disrupted when you when you bring it water to it, or an OH bond of some kind. So the chemistry is quite different when you disrupt that intramolecular hydrogen bond. And so just haven't followed up with looking at the transition states. Thank you.
The next question is from Gernot Friedrichs, who asked a very interesting question, actually. The question is, what is the predominant class of chemicals found at the environmental air-water interfaces? Does it include a high fraction of photoactive compounds, or is there a small amounts of photocatalysts that drive the photochemistry?
Yeah, it's a very interesting question, and probably somebody like Kim Prever and people in CASE could answer that better than I who do the field measurements. But I think part of what you will see is a mixture. And so this is what actually inspired our looking at mixtures where you have some photocatalysts, but also some inert species. What will be at the surface will be, everybody, the surface of the ocean is basically a giant separator. What will live there will be surfactants, but even some soluble species. And then the chemistry can be quite promiscuous, because you don't need much of an activator. So it just so happens that these oxoacids are great because they absorb in the current solar spectrum, while fatty alcohols and fatty acids absorb in the VUV, which is not present. Some of these could have been reached in prebiotic times when we had more UV. So we've considered some of that chemistry that would be quite different. But currently oxoacids are pretty good at promoting photochemical reactions. But for a list of what's there, I've looked at various lists and so forth, but I would like to suggest talking to somebody who does do the field measurements.
Thank you.
Okay, the last question is from Jahan Dalati. He has a question about droplets. Should I think of droplets as just a means of having much larger surface area? Or is there something inherently different about the chemistry of very small droplets, say 1 to 5 microns, compared to a large surface?
So I think it's a very good question. I think if you look at 1 to 5 or so micron droplets, that's true of what we nebulize. It's true of what's being used in the XAIR experiments and so forth. Compared, the droplet size is huge compared with the molecular dimension. So to a molecule, the droplet is flat. So we'll have to go to much, much smaller objects to worry about the effect of curvature. We right now are in a regime, which happens to be the most likely aerosol size as well, where there isn't much of an effect from the curvature of the droplet. So, so indeed, what you gain is you gain quite a bit of surface area. And the microdroplet work, which is not my own, Graham Cooks and Big Zaire and Kevin Wilson and so on, have all come to the conclusion that they see this peculiar enhanced chemistry. It's actually a surface effect. It's not due to some of the other possibilities that would be confining the reagents in the interior. So they appear to be interesting because they have a large surface area.
Great. Thank you so much, Veronica, for
You're welcome.
the talk and